Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 160th seminar, and I call it University of Delaware Day. This week, many things happen. Pope Francis, who significantly changed the conservative Catholic, passed away, unfortunately. Regardless of religions, it is a huge loss, a tremendous loss of a great world leader. In this challenging time, we need great leaders like him. Personally, I visited Cleveland Clinic to give a department seminar. It was meaningful to me because the first place that I visited in the United States in 2003 or 22 years ago for the first time was Cleveland Clinic. And what a interesting coincidence. At that time, I attended a conference as a researcher at a biotechnology company. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Kelvin Lee. He is the Nimble Institute Director and also Gore Professor of Chemical and Biomolecular Engineering at the University of Delaware. As you may know, Nimble is a huge research center that aims to advance science and technology through collaborative research and interdisciplinary training. He is a truly great leader, making this important mission achieve, of course, through collaboration. And Calvin, thank you so much for your great leadership and contribution to the research community and education. The virtual podium is all yours now, and thank you again.
Thank you very much for the opportunity. I'm sharing my slides so you can let me know if you're not seeing them. But I really appreciate the opportunity to spend some time with all of you. I wanted to send as a theme in my few minutes here with all of you to talk about the importance of maybe collaboration and how I got to where I was in terms of learning and experiencing the value and the benefits of collaboration. So that'll be a little bit of a theme, but just to reflect back onto earlier in my career, I'm for better or worse of a vintage where when I started graduate school and my training is in chemical engineering, there were really almost no chemical engineers doing any molecular biology. There was maybe only one at the time. But it was also the dawn of the biotech industry as it relates to biopharmaceuticals. And a lot of that early work that got pioneered in bringing life-altering, life-saving medicines to patients, a lot of that work was really developed at the interface of academics working in collaboration with industry to develop those processes, to do the cloning and the transfection of those cells and ultimately bring products to market. So I'm going to briefly just talk through three very short 2 to 3 slide vignettes on things that have been meaningful to me to work on. And the first is related to the CHO genome. So the CHO cell, the Chinese hamster ovary cell, some of you will know is the workhorse. It is the predominant host cell that's used by the biopharmaceutical industry to make hundreds of billions of dollars US of products every single year, recombinant proteins, therapeutic antibodies, and so on. And about around 2010, within the community, there was, we were looking around and seeing that every other organism and community seemed to have a sequenced genome of substance and a reference genome to rely on, but the CHO community didn't really have that kind of resource in place. And we talked as a community about the value of having not only a reference genome to build from, but also having a place to share that information. That having that kind of sharing of basic information could help really improve everybody's understanding, whether you're in academic, whether you're in government, or whether you're in industry, basic understanding of CHO cells. It could be a way to promote and foster collaborations. And it could be a way to also support the needs of the industry at the time. So we did a couple of things along with others. We first established a website and a community, and you can see the logos of the organizations that help contribute to that, whether it was funding or direct work and advice on how to do that. We built a website, and then around the same time, we also started working with others in thinking about how we develop a sequenced genome. And part of that was a conversation about how do you get a mammalian genome sequenced and funded, and who's going to do that work. And today that almost seems like a trivial question with today's technology and how quickly the costs have come down. But back at this time, it was a non-trivial question. And so these were the days of Kickstarter as a new kind of phenomenon. And so we thought about a crowd-funded effort or a community-funded effort to update and refine reference genomes and bringing together individuals from across a range of organizations to help do that work. And that those contributions are celebrated with this particular slide. So I'll condense the entire history as I understand it into this one slide with a series of landmark publications. CHO K1, the cell line, that genome was published first in 2011. That was the first ever reference sequence for the community. But as people continued to collaborate and see the value of sharing, there were then Chinese hamster genomes that replaced, in some sense, the CHO K1 genome as the benchmark in 2013. And in 2018, a significantly updated and improved genome, which goes by the acronym of PICR. That is a meta-assembly of short and long read technologies that closed 95% of the gaps that previously existed, and where 90% of the genome sequence was in the top 122 scaffolds. And then a few years later, using some more modern techniques, we're able to get chromosome scale scaffolds, that is one scaffold sequence of chromosome length for every chromosome, and 97% of the genome placed onto scaffolds, to the point where, by most metrics, the reference genome for the biotech industry now going forward is almost equal in quality to what you have for human and for mouse. And that really has been enabling for the community, and it's only come about through collaboration. One of the other things that have happened maybe about 4 or 5 years after we started that genome community was then to think about ways that the community could collaborate between universities and the industry. And so some colleagues and I imagined what it would be like to create a National Science Foundation, NSF, funded Industry University Cooperative Research Center, and we created something called AMBIC, which is the Advanced Mammalian Biomanufacturing Innovation Center. The way these centers work is you have a collection of university partners, and you can see the logos here led by Johns Hopkins University, and then you have a number of industry partners who come in, and the goal is to really try to shape and define pre-competitive research challenges that are of interest to everybody. So we see AMBIC as a way to advance biomanufacturing technologies, to enhance competitiveness of the industry, and foster educational opportunities for interested students. And one of the living mission statements that we have for AMBIC is that it's fast-tracked advanced biomanufacturing focused on industrially relevant biology, trying to understand the biology of CHO cells, relying on systems biology and synthetic biology approaches, how we think about process monitoring and control, and how we think about consensus and standardization issues. And I'll just share with you the logos of the organizations that have been collaborating inside of AMBIC as of fall 2024. It really is, I think, a pretty diverse group of companies representing drug product manufacturers, suppliers, and as well as even government agencies. The last thing that I'll point to, which is even a larger scale type collaboration, is nimble and industry, academia, government collaboration designed to accelerate innovation and solve industry challenges and develop a skilled workforce. Nimble is a little different than AMBIC in that Nimble takes technologies that are already proven, so it isn't so much about research, but it's around de-risking technologies so that industry will adopt new manufacturing approaches and new manufacturing paradigms. It is really about trying to advance the field as a whole. And it's not only about cell line, it's about purification, it's around final formulation, it's around workforce. And so it's really broad in scope. And the Nimble mission, very briefly, is to accelerate biopharmaceutical manufacturing innovation and support the development of standards that can help everybody have more efficient and rapid manufacturing and educate and train a world-leading workforce. We are heavily supported by the US government, so there's an obvious focus on trying to promote the needs of US competitiveness in this industry, but it certainly is a global industry. There's manufacturing happening all around the world to benefit people all around the world. And just the one last, apologies, a little bit complicated slide on this. When we thought about Nimble, we thought about what the needs are in terms of US competitiveness, around workforce access, around the rise of new classes of medicines, precision medicines, the value of standardization. And we bring together within Nimble a range of stakeholders across focus areas and process themes, ultimately trying to help the industry achieve some of the outcomes that it seeks around continuous manufacturing, novel real-time analytical tools, energy and water savings, ultimately helping the industry move faster, more efficiently, and having the impact we all seek, not only on economies and supply chains, but certainly also the public health benefits to all. That was really kind of what I wanted to share. It's been a really kind of interesting journey for me, thinking about the value of collaboration and what we can all accomplish when we work together. And I've really benefited from the opportunity to not only learn from those who came before, but also partner with those from all different fields and all different stages of career. And so with that, I thank everyone for your attention and listening to my remarks. Thanks.
So thank you so much. That's fantastic. I really like you kind of giving us the history of cell, field. I didn't know. I mean, the even sequencing is not available until 15 years ago or 14 years ago. That is a very interesting point. But I also do see the power of international collaboration that is basically done by international collaboration to make that information shared and sequencing done. And that's fascinating. Also, as a synthetic biologist, I believe this field is around 25 years old, but I do believe this is a time to deliver real product. But your, effort is already kind of generating a lot of product by real collaboration between, industry and academia, also the government together. So that's a fascinating effort. And thank you so much for this amazing, leadership and also the contribution to the society. Thank you.
Thanks.
All right. So now the main speaker of today with a longer introduction. Dr. Haosheng Shen is a research scientist at the National University of Singapore, or NUS. He earned his bachelor degree in biological science from Nankai University, China, 2016, and completed his PhD at NUS in 2021 under the supervision of Professor Matthew Chang and also Professor Yongshan Lee and Professor Wenshan Yu. Specializing in microbiome engineering, he focuses on the use of engineered commensal Lactobacillus strains for targeted therapeutic delivery in treating disease. Conventional chemotherapy, while remaining a standard approach for cancer treatment, is often hampered by poor bioavailability and severe systemic toxicity. I know this fact because my dad also suffered from the toxicity, and I actually lost him last year. Prodrugs have emerged as a promising alternative for targeted, cancer therapy. Despite this, current prodrug strategies face limitations in target specificity and complex pharmacokinetic profiles, which can affect their distribution, metabolism, and clearance within the human body. To overcome these challenges, his research focused on leveraging the intrinsic interactions between bacteria and cancer cell within the tumor microbiome for precision or precise prodrug administration. I hope this work will overcome challenges associated to cancer treatment, helping many, many patients suffering from this global disease. Haosheng, thank you so much for your time today, especially at the very inconvenient time for you, I believe past 11 p.m. your time. And please take it away, and all yours now. Thank you.
Thank you. Thank you, Prof, for the nice introduction. And I'm Haosheng from National University of Singapore. I'm working with Prof Matthew Tan in development of lactobacillus-based autonomous nasal drug delivery systems. So I'll start with a little introduction of the Tan lab. Our lab is deeply interested in designing genetically encoded circuits that enable living cells to perform new cellular functions. We are studying how we can engineer the genetic circuits with a more systemic and predictable manner, and how the living cells will respond to these newly engineered genetic circuits. And at the upstream of our research, we designed the entire microbial chromosomes and constructed the redesigned chromosomes from scratch. This bottom-up genome engineering methods tells us a lot of interesting features in the microbial chromosomes. And at more downstream, we engineer genetic circuits to give new cellular functions to living cells. These engineered living cells could perform like bacterial cell factories and also even living therapeutics. And this is the topic that I will dive into today. So the engineering of autonomous bacterial drug delivery system in our lab are built around the host microbial interactions, and the engineered microbes are engineered. The microbes are engineered either to target specific structures of the host cells or to respond to host inputs from disease conditions to enable in situ, on demand, and targeted drug delivery of therapeutics. As we all know, the human body is a huge reservoir of commensal microbes, with all these commensal microbes interacting with our complicated metabolites in various organs and tissues. And my projects are mainly focused on the nasal cavity and the interaction of a commensal bacteria called Lactobacillus plantarum and its interaction with a polysaccharide molecule called heparan sulfate. So heparan sulfate is commonly expressed in a lot of human tissues, but in the nasal cavity, it's exclusively expressed in only two places. And the first one is the nasopharyngeal carcinoma tissues, and the second one will be on the apical layer of the olfactory mucosa in the nasal cavity. I will start with the project that tells the story of how we use the plantarum to do targeted delivery for cancer therapy for nasopharyngeal carcinoma. So Nasopharyngeal carcinoma, or NPC, is the most common head and neck cancer and has a very high incidence rate in South China and Southeast Asia. And like many other cancers, nasopharyngeal carcinoma are reported to host a range of different commensal bacteria inside the cancer. And more interestingly, for the case of NPC, both the healthy nasal cavity tissues and the nasopharyngeal carcinoma tissues actually harbors a list of commonly known probiotic strains called Lactobacillus. So this actually led us to formulate if we can use a hypothesis that if we can use these commensal bacteria for the delivery of targeted chemotherapy reagents from the nasal cavity directly to the nasopharyngeal carcinoma to reduce the systematic exposure of chemotherapies, which were usually given intravenous systematically. So the first step we do is to try to isolate an engineering chassis that has a very high active interaction rate with the NPC cells. And then we use the NPC cell line called CNE1 and screen that list of bacteria, commonly used commensal and probiotic bacteria strings. And we find this Lactobacillus plantarum cells has a significant higher binding capacity towards CNE1 cells compared to all the other probiotic strains. And then we quickly used this same plantarum strains on a lot of other NPC cancer lines, and then find that the bacteria human cell binding is actually very specific to cancers and quite absent on a normal nasal epithelial cell line. So when we added heparin, sodium heparin in the co-culture of the bacteria and cancer cell lines, we find that the addition of the heparin actually blocked the binding of the bacteria towards the cancer by up to 50%. Sodium heparin is the analog of heparin sulfate, which is commonly presented on the surface of cancer cells, and this led us to think that heparin sulfate could be an important component for the bacterial cancer binding. Then we check the literatures, and then we find the Lactobacillus species actually express a lot of oligopeptide substrate binding proteins on its membrane, which are known to have inherent heparin sulfate binding capacity. And in this plantarum cells, there are 7 of these OPPA proteins, and we stimulated the structure of these proteins, and then perform the heparin docking on all of the proteins, and then find that amongst the protein, this LP0018 protein has the strongest binding capacity towards heparin. And we purified all the heparin OPPA proteins and then incubated with all the NPC cell lines, and then find that aligned to the simulation results, that LP0018 actually has the strongest binding capacity towards all the NPC cell lines, which makes us wonder if heparin sulfate is indeed the key binding component in this bacterial cancer interaction. And then we want to further validate the role of this heparin sulfate binding protein in the cancer bacterial interaction by directly adding the recombinant LP0018 protein in the co-culture of the bacteria and the cancer. In doing that, we were hoping that the bacteria the protein would actually compete with the bacteria over the same binding sites on the cancer, and the addition of the protein would reduce the binding of bacteria towards cancer. However, this did not happen, and on the contrary, the addition of the heparin sulfate protein increases the binding of the bacteria towards the cancer by 8 to 20 folds in various NPC cancer cell various NPC cancer cell lines. And then again, the addition of the heparin salts completely demolished the enhancement of this cancer binding. So we double checked the structure of the OBPO protein and then realized that it's actually is actually a lipoprotein which would actually binds to both the bacteria the bacteria cells and then the cancer cells. We verified that the binding of this LP018 protein towards the bacteria, and when we truncated the N terminus of this protein, the binding towards the bacteria is demolished, and then the enhancement of the bacteria cancer binding is also demolished in a co-culture, and this truncated LP018 protein actually, on the contrary, blocked the binding of bacteria towards cancer. So by far, we have covered that the Lactobacillus plantarum strain could bind specifically to the NPC cancer cell lines via the recognition of heparan sulfates through its oligopeptide proteins on the membrane. We want to further utilize this bacteria for a specific targeted therapy for nasopharyngeal carcinoma, and we did it by engineering the bacteria to surface display a streptobaiting protein on the bacteria, and the streptobaiting protein on the surface of the bacteria would enable the loading of biotinylated chemotherapy prodrugs, where these prodrugs are engineered to have this reactive oxygen species active linkers, which could be activated when the whole compound is close to the NPC cells, and then release SN38 as the chemotherapy reagent and kill the cancer. So we engineer the bacteria to both secrete and then surface to display streptobaiting protein, and then as a result, a multimeric form of streptobaiting is displays on the surface on the bacteria, and were actually detected through Western blots, immunofluorescence staining, and also flow cytometry. We also designed this Prodrug based on a chemotherapy reagent called SN38, and as you can see, the structure of the Prodrug consists of 3 different parts. Biotin in orange on the left, and then SN38 in green on the right. And in the middle, there's a linker that can actually sense redox signals or glutathione signals coming from the cancer cells. And once they are activated by the redox and glutathione signals, they will release SN38 from the compound, and also release SN38, then release the SN38 from the surface of the bacteria. So we went on to test the efficacy of this strategy first in the cell line, in the NPC cell lines. We co-cultured, we added prodrug loaded bacteria into the NPC cell line culture, and then we find that the 24 hours after co-culture, the SN38 could be actually released from the bacteria surface and gets accumulated in the NPC cells. And then we also tested the killing efficacy of the strategy towards the NPC cell lines, and we find that surprisingly, the prodrug itself did not enhance any killing of the cancer cells, but the prodrug loaded bacteria could actually reduce the IC50 of the chemotherapy reagent, the SN38, from by 5 to 10 folds in various cancer lines. Then we went on to test the efficacy of this killing strategy in a mouse xenograft model. First, we planted the tumor on the back of the mouse and then we give the bacteria intravenously and then try to see if the bacteria can actually colonize in the tumor itself. So we find that 24 hours following the intravenous administration, the bacteria are predominantly found in the tumor, but quite absent in most of the other organs, and the colonization of the bacteria actually persists within 72 hours. And very interestingly, we double check that the colonization of the bacteria through the CFU counting and find that actually 24 hours following the administration, there are still a lot of bacteria in the liver and spleen, most likely being captured in neutrophilic cells. But 48 hours following administration, most of the bacteria are actually cleared from the non-tumoral organs, which compared to most of the bacterial-mediated cancer therapy, this is a very fast clearation, and then indicating that the lactobacillus are actually a very safe chassis for bacterial-mediated cancer therapy. And also in the serum, the bacteria was no longer detected 6 hours following intravenous administration. We used another lactobacillus strain which has a lower binding in vitro to cancer cells, and then compared to plantarin compared to plantarin, it also shows minimal colonization of minimal colonization in the tumor. We then tested the efficacy of the prodrug loaded bacteria in the cancer model, and then we find that after 4 doses of intravenous administration, the bacteria could significantly slower down the growth of the tumor and showed a very decent inhibition on the tumor. And this work was published last year earlier in Nature Com. And after that, I'm going to share another story where we use the Lactobacillus plantarum for targeted delivery of targeted nose to brain drug delivery. So as we all know, because of the existence of blood-brain barrier, a lot of to deliver drugs from the blood to the brain is actually very difficult because the BBB would impede the delivery of most of the therapeutics from the blood to the brain, from the blood to the brain. But as we also know that one of the very important function of our nose is its function of olfaction, and the olfactory mucosa in the nasal cavity actually connects directly the nasal cavity to the central nervous system, and the olfactory epithelium actually serves as a perfect target to do intranasal delivery to deliver therapeutics bypassing the BBB from the nose directly to the brain. However, the difficult part is that in the human nasal cavity, 95% of the surface area is respiratory epithelium, while the target epithelium, the olfactory epithelium, takes only 5% of the surface area, which means that if we want to do very efficient intranasal delivery, we need to provide a strategy with very good olfactory targeting. And on the other hand, there are a range of commensal microbes in the nasal cavity because nose is a semi-open environment, and then amongst these nasal commensals, we can also see a very well characterized probiotic species called Lactobacillus. So the question is, yes, we need a better targeting, OE targeting for efficient intranasal delivery, but can we use these commensal bacteria for the targeting of olfactory mucosa and then use these bacteria as the chassis for nose to brain drug delivery? And the answer is yes, because again, there are this molecule called heparan sulfate, and is more specifically an acetyl heparan sulfate that's exclusively presented on the apical layer of the olfactory mucosa. So this led us to hypothesize that we can use the commensal bacteria in the nasal cavity like Lactobacillus to target an acetyl heparan sulfate and to target olfactory epithelium for targeted nose to brain drug delivery. And again, the first thing we do is to screen a proper engineering chassis for the delivery of the drug, and then we used a list of commonly seen commensal cells in the nasal cavity, and screened those bacteria against the binding towards the NS0 cells, which expresses N-acetyl heparin sulfate on the surface. And again, we find the same L. plantarum strain with a very high binding towards NS0 cells. We then tried to do a pull down to identify the specific protein that's responsible for the binding towards N-acetyl heparin sulfates. And again, we identified a OPA protein called LP0783, which belongs to the OPPA protein family. And then we used the two cell lines to specifically check the binding of the OPPA proteins towards the N-acetyl heparin sulfate, because we want to know which proteins amongst the OPPA proteins are actually responsible for the binding of N-acetyl heparin sulfates. We use the dual K1 cells, which expresses all type of heparin sulfate on the surface, and NS0 cells that expresses only the N-acetyl heparin sulfate on the surface. And then we find that these three OPPA proteins has a significant higher binding towards N-acetyl heparin sulfate compared to the rest of the proteins. And we think that these three proteins might be in charge of the N-acetyl heparin sulfate binding. And following that, we hypothesized that if we can use the bacteria to target the olfactory epithelium through the N-acetyl heparin sulfate binding, and use it as a delivery chassis to deliver therapeutics from the olfactory epithelium to the olfactory bulb and further structures in the brain. So the experiments we do following this is quite straightforward. We labeled the bacteria with a green fluorescence dye called FITC, and administered the labeled plantarum cells into the nasal cavity of the mouse, and then tried to check where the bacteria actually localize. And here on the left upper panel, we can see the rough location of the respiratory epithelium and olfactory epithelium in the nose of the mouse, and also olfactory bulb in the brain of the mouse. And on the right, we can see the panel of bacterial localization after intranasal administration, and we can see that 24 hours following intravenous administration, we can tell from the IVIS that the bacteria are majorly localized in the olfactory epithelium in the nasal cavity of the mouse. And when we deleted 2 copies of the OPA protein that binds to N-acetyl heparin sulfate, the localization of the bacteria reduces roughly about 50%. We can tell from another angle that the bacteria are actually predominantly localized in the olfactory epithelium following intranasal administration, meaning that the bacteria could actually binds to targets to the olfactory epithelium. We then performed a thorough section of the mouse nose, and then tried to find the exact location of the bacteria. So you can see from the right panel, we start the section from the left panel, we start the section from the respiratory epithelium, and we can see that following up to 14 days of consecutive daily administration of the bacteria, we don't see any residual bacteria inside the respiratory epithelium. And when we section deeper, we used an antibody against the olfactory marker proteins to identify the olfactory epitheliums in the mouse nasal cavity. And following 3 days of consecutive daily administration of FITC labeled plantarum, we find a predominant accumulation of FITC beneath the in the lamina propria beneath the epithelium of the olfactory epithelium of the mouse. This could be that the FITC molecules are gradually released from the bacteria, and the released FITC are actually accumulated in the tissues connected near the presence of the bacteria, and hence, because the bacteria specifically localized into in the OE, the FITC is predominantly accumulated in the lamina propria beneath the OE. In the deeper sections, we can actually see the accumulation of the bacteria in the OE itself, following 3 days of consecutive administration of bacteria. Yeah. When we amplify the section and take another look into the lamina propria, we can actually see that the olfactory nerve bundles marked by the anti-OMP antibodies gains its size when we go deeper and deeper in the section, and the FITC are actually accumulated around these olfactory nerve bundles, which serves as a gateway for the FITC to diffuse into further structures. And in the nascent olfactory epithelium sections, we can clearly see the accumulation of the FITC after 3 days consecutive administration of the bacteria. But in deeper sections, we can only observe a few diffusion of the FITC from the olfactory epithelium slowly to the olfactory bulb in the brain, like 7 days following the administration of the bacteria, which possibly means that the diffusion of the FITC requires certain concentration, which also requires a further longer administration time and accumulation. And in corresponding to these sections, we find accumulation of FITC in the olfactory bulb in the mouse brain following consecutive intranasal administration of the bacteria in the nose. And from another angle, we can actually see that the FITC are diffused to other parts of the brain and quite widespread to the other structures in the brain. This means that the plantarum cells could actually deliver its payload molecules to the olfactory epithelium, and then following its accumulation in the olfactory epithelium, the molecules could be released in the OE and gets diffused into further structures and eventually ends up in the brain. So to prove the concept of to prove the concept that we can use the bacteria for the delivery of therapeutics, we engineered the bacteria to secrete three human hormones, human appetite regulating hormones: alpha, MSH, BDNF, and leptin. We verified the bioactivity of these recombinant hormones, and then administer these engineered microbes into the mouse, and then which are under high-fat diets, and then try to observe if these engineered microbes could provide a preventive effect on obesity. So we did another section in the mouse nose, and then find that after the administration of these engineered microbes, the bacteria can actually release the hormones into the nose, and then the hormones are actually diffused through the OE to lower structures into the lamina propria, and then probably eventually reach the brain. And more importantly, the bacteria, the administration of these bacteria could actually prolong the existence of these hormones in the nasal cavity to 24 hours to 48 hours. But because which is quite important because the half-life of these hormones themselves are actually only like lesser than half an hour. So the administration of the engineered microbes actually largely prolonged the duration of these hormones' existence in the nasal cavity. Yeah, and then we went to we did a consecutive administration of daily administration of these engineered microbes in the mouse for 8 weeks. The mouse are put under high-fat diets, and then we monitored their body weight gain for consecutively for 8 weeks, and then we find that at the end of the treatment, all of these engineered microbes were shown to reduce the body weight gain of the mouse, reduce the daily food intake, and improve the glucose metabolism and the fat deposition, meaning that they provided a very positive preventive effects on preventing the obesity in mouse. And they also reduced the liver cirrhosis from forming in mouse under high-fat diet. And we did an extra experiment by directly comparing the bacteria, the engineered microbes to recombinant hormones. Because in the previous model, bacteria that excrete leptin yields the best effects in preventing obesity. So we directly compared the bacteria that secretes leptin to a recombinant pure leptin peptide bought from industrial sources. And very interestingly, following the intranasal administration of the recombinant leptin peptides, we don't actually observe the presence of the leptin 24 hours following intranasal administration. While compared to the engineered microbes, engineered microbes could actually prolong the existence of these leptin in the nasal cavity to at least 48 hours. We then compared the effect of the recombinant leptin and the engineered microbes in preventing the obesity in mouse by through our animal model, and then we implemented two groups here where the bacteria or the recombinant leptin are given either daily or on alternative day. And it's interesting that quite interestingly, we find that when both of them are giving on the daily dosage, they showed a similar effect in reducing the body weight gain in mice. But when they are given in lower dose on alternative days, the recombinant leptin failed to show any protective effect against obesity anymore, whereas the engineered microbes still performs the same way as the high dose leptin, which means that our microbes can actually largely prolong the existence of the hormones in the nasal cavity and then prolong the therapeutic effect through the intranasal delivery, which in an ideal way, the bacteria could regulate the appetite in obese patients with very little dose, with very little bacteria given and very less frequent bacteria given, which is quite beneficial in preventing obesity. Yeah, as a summary, we implemented the development of autonomous therapeutic system based on lactobacillus in our lab, based on three gradual steps. First, with inherited first with the analysis of the inherited targeting property of the lactobacillus in the first we did the analysis of the inherited targeting property of the lactobacillus to different structures in the human nasal cavity. And then we employed different engineering strategy in both cases to use suitable genetic tools to deliver the therapeutics. And then we performed the proof of concept delivery of the therapeutics for the treatment of the disease. So in the engineering of lactobacillus are actually quite difficult, and then the two projects actually took me 9 years in total, like to publication. But this gradual systematic way of engineering benefit me in multiple ways, including generating a large library of basic parts, and provided a lot of information of the interactions of the bacteria towards other tissues or organs in the human body that could actually benefit me in my future working. And finally, I want to thank all the group members that were spending all the time with me in the lab for like consecutively for like 9 years, and then I really appreciate their support and help, and also all the funding sources that supported us for the past few decades. Yes, and that will be all of my presentation. Thank you.
Fantastic. Of course, the big achievement takes time, and 9 years is
Yes.
long, but with this huge achievement, I believe your 9 years is worth it, I guess. So
Thank you, Dave.
let me start with some question. So
Okay.
I'm fascinated by your second part of the talk. Especially it's kind of very unique or novel, also clever idea to deliver drugs to the brain using, nose.
Thank you.
I wonder what would be, characteristic of the drugs in terms of size, or hydrophobicity, or etc. to deliver into the brain. I believe there should be some limit, but I just wonder what would be that. I probably
Yes.
missed
Yes. Thank you for the questions. It's a very good question. So yes, there will be a lot of limits, and the major limits will come on the based on the property of the drug, because uh we are actually just doing a delivery tool using the bacteria. We're just using the bacteria as a delivering vehicle to deliver the drug from the nose to the olfactory epithelium. But if the drug itself could not pass the olfactory epithelium, or in other words, if the drug could not be delivered intranasally to the brain, then our bacteria couldn't actually do any help to facilitate the delivery of the drugs. Which so an intranasal delivery of the drug is actually a very big topic, and then people have actually characterized a lot of different drugs that can be delivered intranasally. And that's why when I started this project, I picked leptin, alpha message, and BDNF, these three hormones, to do a proof of concept, because we know that these three peptides are actually able to be delivered intranasally to the brain. Yes. Yeah. And that will be the major obstacle for the payload molecules, for the choice of the payload molecules. Yeah.
So,
But then again, yeah, sorry.
no, go ahead.
Yeah. But then again, we already have a lot of information because people have already done a lot of intranasal delivery studies to study which molecules could actually be delivered. Yes. Yeah.
So but do we know let's say molecular weight upper limit for that drug? Because I do remember many years ago we need to get the vaccine for, I believe, what was that? But there are one option for the kid, including my daughter, they
Mhm.
don't like, neither. And then,
Okay.
but he got the nasal vaccine, and then
Yeah.
somehow that is not going, of course, to the brain. That's probably going
Yeah.
into the
Yeah.
other organ or blood. So that means, I mean, the potential of this method or this approach is huge. But probably still kind of there should be some upper limit of the size
Yeah.
of the molecule or some
Yeah.
property, etc. And I just wonder what is the upper
Yeah.
limit.
The boundary for the upper limit of the size of the protein is quite blurred, I would say. But
Oh.
in general, large protein or large molecules or hydrophilic molecules doesn't really go well in intranasal routes to the brain.
So it could be
Hydrophobic molecules, yeah,
protein and also small one. That should be
yeah,
the target, you
yeah,
need to
yeah.
deal with, right?
And also delivery from the nose to the olfactory epithelium is not actually so easy because olfactory epithelium for human are at the very top nasal cavity here, and then there are a lot of obstacles for the molecule to actually pass from here to here. So usually when the drug is given to olfactory epithelium, the person has to actually have to like stand upside down and have an
Uh,
impulse, and then the drug will actually go slowly into the topmost part.
uh-huh,
Yeah. So as the case as you were mentioned, the nasal inhaled vaccination, it will probably won't reach that space so far in the nose,
uh-huh,
and it will mostly go through the olfactory, so go through the nasopharynx
uh-huh.
region where there's a very large lymphoid nodes, and then which can generate immunity against the vaccine.
So depending on where your drug want it to be,
Mhm.
I mean, mouse
We,
experiment you probably done in that way upside down and
yeah,
then
yeah, yeah,
forced
yeah.
to
I
deliver.
just hold it and then. But
Got it.
for human, I guess we need a lot of additional tools to
Uh,
help the administration. I think,
So
like
vaccine is okay.
yeah,
Vaccine is okay to deliver by nose because that's the immune response. That's what we need to have.
yes, yes, yes,
But
yes.
other drug might be some limitation for human.
Yes,
Uh,
yes. That's also one of the reasons why we want to use the vector to target it and just to stick on the olfactory epithelium and continuously secrete
uh,
therapeutics. Yes.
that makes sense.
Mm.
One last question. Let me think. Let me see whether we have question from audience. Okay, I don't see
Okay.
any. So one last question is, this is because of my interest. This is the so-called GMO. You just
Oh,
generate.
yes,
And then how you gonna deal with GMO biocontainment issue? I believe
yes,
Asian country, also European country not like GMOs.
yes,
So the
yes.
regulation that should be something you need to consider.
Yes. So yes, people kind of just freak out when it comes to GMO, and I believe the FDA has some rules for the application of these GMOs, in which the first thing is that the plasmid systems cannot be used, and all the genetic modifications should be done directly on the genome itself. So it reduces the chances of horizontal gene transfer and pollution of those genes. That will be a very important thing if we want to push this into translation. And in the future translations, we are also actually looking into modifications that does not require engineering of the microbes. Like we
Mm.
use surface modifications on the bacteria to deliver designer molecules, or we engineer the microbes into other like smaller cells like mini cells, which doesn't have the chromosomes anymore. And then so apparently then we can't really call it a GMO because it's just basically recombinant cell without any DNA. Yeah. And we will try to like go through these different strategies to avoid the use of these traditional engineered microbes to do the research, because then also the bacteria will be safer to use and then doesn't have antibiotic resistance. And like let's say for mini cells, it doesn't even replicate anymore. So it will be our more convenient and disposable vector for delivery of therapeutics. Yes,
Sure,
that's what I have
absolutely.
so
That's
far.
the great, answer. I also know, I mean, synthetic cell community also try to use non-replicating system to
Yes.
kind of deliver, those kind of the drugs. Also using the cell-free system as well. So that's fascinating. Okay, let me see. I don't see any question from audience. Also, this is already past your midnight,
Okay.
so I need to let you go. So just
Yes.
close. Okay, so
Thank you. Thank you, Prof.
yeah, thank you so much for joining and staying today. We'll meet again on May 1st. Thursday, the same time, the same Zoom link. We'll have
Perfect.
Professor Cesar de la Fuente at University of Pennsylvania and Dr. Nika S. Tilly at J. Craig Venter Institute. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks.
Okay.
I stop recording. Just 1 second.